Hello YouTube, today I've been given this DNA sequence and was tasked to make a 4-long amino acid chain from this. Looking at the first one, while we have something that will equate to a start codon, we can't find anything that is a stop codon. So I took the second one and rewrote it into the 5 to 3' direction. Now we go through a translation and take this DNA and make it into RNA. Now we can see the start codon here, and in the 5th position, UAA, it would have been a stop codon. Now we go through transcription and make it into amino acid chain, and we would have methionine, valine, serine, lysine, and then we would have the stop. The stop doesn't actually transcribe into a amino acid. If during replication of the original strand, something would have went wrong. For example, this A would have changed into a T. This would have also changed our RNA, and through this it would have also changed our amino acid end product. But as the DNA changed to a T here, this would have also changed into a U instead. And as we went from a purine, from the A originally, to a pyrimidine, the T, the thymine, this would have not coded for the stop here, but instead would have coded for leucine next, then serine, and then thymine. This would have made it into a long, non-functional protein, and as I said before, this was transversion. Another one would have been nonsense mutation. A nonsense mutation is when we have an earlier stop codon. So in this case, during replication, once again, something changed into the coding strand, and let's say, for example, this A instead would have coded for a T. This would have been a transversion as it went from an A to a T, from a purine to a pyrimidine, and then the amino acid chain would have changed, and this would have changed into UAG, which is a stop codon. So it would have only been 3 amino acids long here. We'd also have missense mutation. We would have an ASN here in the S9 instead of the original lysine, which it should have been. This has changed the amino acid, which may have altered the function, or it didn't work as a new amino acid. This was transversion, as once again we went from a purine G to a pyrimidine C. Then we have another case, which is called silent mutation. A silent mutation is when it would have, for example here, read for another A, and this would have also read for another A. This would have still been lysine, which was in the original. It wouldn't have changed anything about it. There would be none effect on the protein.